So, an ECG is a graphic representation of electrical activity within the heart, and it's taking place during the cardiac cycle. Now, impulses from the heart spread to other body tissues and fluids that are found near the surface of our skin. And as a result of that movement, we can measure these events through electrodes, and ECGs can pick up distinct waves. Now, when you're looking at this particular image, I'm not going to go through the segments. I'm just going to go through the waves because anything more detailed than what I'm going to go through is really better served in a different type of course. So we're just going to look at, our P wave, our QRS complex, and our T wave. Now, when we are thinking about these different waves, we'll use the terms depolarization and repolarization, and these are changes in electrical distribution within the heart. And when it comes to depolarization, what it specifically means is a discussion for our neurons chapter. But what we're going to think about here is that depolarization is going to change the electrical distribution in such a way that we'll be able to proceed with systole. And then repolarization is a time in which we're going to head back towards a resting state. And so we'll start to associate that with diastole. But what you'll be able to do at the end of the term is link these specific deflection waves with fluctuations or changes that we would associate with the nervous system or with certain channels or changes with respect to ion concentrations. So the P wave is referred to as atrial depolarization. So it's a time when the atria are depolarizing. And this is produced when a signal from the SA node spreads throughout the atria, and atrial systole begins a little after the P wave begins. The QRS complex we see is a really large one, and that's associated with ventricular depolarization. And it's higher than atrial depolarization just due to the thickness of the ventricles and the increased force in which they contract. So that's going to produce a signal. Okay, and so that's going to happen when the signal from the AV node spreads through the ventricular myocardium. Now, we know that the ventricle has the largest muscle mass, so it's also going to generate the greatest electrical current. There is also atrial repolarization, so meaning there's relaxation of the atria happening at this point. But because of how significant the QRS complex is during ventricular depolarization, it's going to mask atrial repolarization. So even though it isn't specifically indicated here, there is both atrial and ventricular depolarization as well as repolarization. And what we see after ventricular depolarization is our T wave, and that's a time in which our ventricles will relax. And when it comes to ECGs, abnormalities or deviations from this expected trend can indicate problems with the conduction system. So it could let us know if there's something wrong with the sinoatrial node or the atrioventricular node or the Purkinje fiber system, or just issues with the cardiac muscle. Two point three is that patterns of blood pressure and flow reflect the structure and arrangement of blood vessels. And all of this means is there is a relationship between form and function. So the vertebrate circulatory system is going to rely on blood vessels that are going to exhibit a close match of structure and function. When it comes to blood vessels, they have basic patterns of organization in that they're tubular and they have a lumen that's surrounded by tissue layers. When it comes to arteries and veins, they have a really similar composition, but they'll just have a varying distribution of those kind of components. So the innermost layer that we'll find is going to be the endothelium, and that's going to be continuous with the endocardium, which we discussed when we went through our layers of the heart wall. And the endothelial cells provide a surface for blood to flow through, and it's a smooth surface, so it helps to reduce any likelihood of friction or turbulence, which can negatively impact the efficiency of blood flow. And the endothelial cells can also produce chemicals. Now, after the endothelium, you have smooth muscle, and you can see that there is a difference between the amount of smooth muscle seen in the artery side versus the vein side. But the smooth muscle helps to control the diameter of the blood vessel, and it therefore can control the amount of blood that flows to organs. So there's going to be a little bit more of a regulatory role that's seen here within the artery side versus the vein side, because remember, the vein side is meant to be our reservoir. It's meant to be collecting blood. So it's more of a passive process than what we see on the artery side. The outermost layer is composed of connective tissue, and the connective tissue helps to support the blood vessels, and it prevents it from overstretching. And it's much more likely that overstretching will take place within an artery just due to the amount of pressure that it needs to deal with as compared to the vein. So we can see there's a difference with respect to connective tissue as well. Now, when it comes to the capillaries, which are seen in the middle, they aren't exactly the same. So we don't see the smooth muscle here. We essentially just see endothelium and arteries lamina. And capillaries really just consist of endothelial cells. And they're joined by tight junctions in a way that they're organized such that it really only permits the movement of erythrocytes or red blood cells in a single file fashion, so like one at a time. But that's beneficial because when we're thinking about the capillaries, that's an opportunity for exchange to take place. So we want to ensure that each of these erythrocytes, which we'll talk about a little bit more in our discussion of blood in the upcoming module, we want to ensure that each of these erythrocytes is able to pick up and drop off what they need. What we also see is that veins will have valves, while we don't see that reflected within the arteries, and that really is just due to the need to prevent blood from flowing backwards in the venous circuit, and that is due to a strong opposition in certain veins like the legs to gravity. So, gravitational pull will want to bring blood down, but we want to be able to distribute that blood back up towards the heart. And so in order to prevent blood from working its way backwards, we'll expect to see valves there. So when we're looking at this particular slide, just think about the relationship between form and function. Think about how these vessels are similar, how they're different, and how the differences that we do see help to support the role that they're meant to play within the cardiovascular system. Here we have a way to think about the way that blood velocity and area and pressure changes as we move through different vessels within systemic circulation. Now, vessels of circulation are going to vary with respect to their total cross-sectional area. So that's the top panel that's here. And these changes can impact the velocity of blood flow through them. And velocity here we see is measured in centimeters per second. And what is evident when we're looking at the top and middle panel is that velocity is inversely related to cross-sectional area. So meaning that when we have a high velocity, we have a low cross-sectional area. So we see the velocity of blood flow change as blood travels through systemic circulation. It's fastest within the aorta. The aorta is proximal to the heart, as well as other larger arteries, and slowest in the capillaries. So what's happening when we arrive at the capillaries? Their total cross-sectional area is significantly increased. And so that's due to the bunch of branching out into these smaller capillary beds or networks. So even though the lumen of the capillaries is smaller than that of the arteries, it's the combined cross-sectional area of the capillaries, which is far greater than that of the aorta. And that increased total cross-sectional area that's seen at the capillary increases the volume of blood that the vessels can collectively hold. And as a result of that, that's going to slow down the velocity because blood's going to be distributed. So let's take a look at the entire portion of the velocity. So when it comes to speed, the cross-sectional area is going to come back down as we transition from the capillaries towards the veins. And blood is essentially converging into these vessels, and that's what allows for the velocity to pick back up again. Now, the velocity is not going to reach the same heights that we saw initially when we were at the aorta, just due to the distance of the vessels, such as the veins and the superior and inferior vena cava, from the initial kinetic energy that's imparted on blood after it leaves the left ventricle. So for velocity, we see that it's up pretty high at the aorta due to its proximity to the left ventricle. It comes down because the total cross-sectional area at the capillaries goes up. So there's a ton of space for that blood to distribute itself into, but then that blood then starts to converge into fewer and fewer vessels, which brings the velocity back up again. For blood pressure, what we see here is a pulsatile state, so meaning that it goes down and up and down and up in a regular fashion. And the peak is generated by ventricular contraction, and that's the systolic pressure. And during diastole relaxation, the aortic valve closes. That's going to prevent blood flowing back into the heart. And the walls of the aorta are going to kind of recoil. And there's still enough pressure to keep blood flowing. Okay, so we have closing of the aortic valve, pressure response that is lower than what is seen during ventricular contraction, but still enough for blood to continue flowing. So there's going to be that small drop in aortic pressure, and that's the diastolic pressure. And because the aortic pressure is fluctuating here, the mean arterial pressure, or the mean pressure, which is what is shown here, is what we utilize as our kind of quantitative value for propelling blood through the tissue. So the solid line constitutes the mean pressure, but in actuality, that pressure is oscillating as a result of systolic pressure and diastolic pressure. Now, when it comes to the movement of blood as we go from aorta to superior and inferior vena cava, when we're thinking about going away from the heart and then coming back towards it, we can see as we transition from the left to the right that our pressure goes down and remains down. Now, when it comes to movement of blood from one place to the next, we are still relying on the presence of a pressure gradient. So even if the value is low, as long as there is a pressure gradient seen between one vessel to the next, we're still going to be able to have blood flow in a particular direction until it enters back into the heart. Now, in thinking about blood pressure, we know that it's pulsatile. We know that it changes as we transition through different vessels, but there are also different mechanisms that can impact blood pressure, and that can help to achieve homeostasis. And we'll go through specific examples of blood pressure regulation when we get into the osmoregulation portion of our semester, but there are multiple ways in which blood pressure can be regulated. But the means in which we can achieve that are through changing the diameter of arterioles. So we talk really generically about this idea of vasoconstriction and vasodilation in our earlier discussion. But when we say constriction, what we're specifically saying is that there's going to be contraction of smooth muscle, and that contraction of smooth muscle is going to change the size of the lumen. And as a result of that change in lumen size, there's going to be an increase in blood pressure. Now, for vasodilation, there's relaxation of smooth muscle within the arterioles, and as a result, there is once again a change in lumen diameter, and that change in this scenario is going to cause blood pressure to fall. So, contraction of smooth muscle increases blood pressure, relaxation of smooth muscle decreases blood pressure. So here we have a smooth muscle relaxation pathway, which gives us an opportunity to revisit the ideas that are found within Chapter 11 when it comes to cell communication and signal transduction. So in step 1, we have acetylcholine, which is our ligand, and it is going to bind to a receptor that's found on the endothelial cells of blood vessels. Now, that ligand serves as our primary messenger, and as a result of that binding event, it's going to activate the receptor, and that's going to result in production of IP3. Now, IP3, we know from the cell communication chapter, is an example of a second messenger. So IP3 is going to be a ligand that can bind to a ligand-gated channel that is seen within the cell. So we have an IP3 gated channel The binding of IP3 to that channel is going to allow for calcium ions to leave the lumen of the endoplasmic reticulum down their concentration gradient out into the surrounding fluid, so essentially out into the cytosol. So again, we have acetylcholine binds to its receptor. That receptor binding leads to IP3 production, which is a second messenger. IP3 will bind to a channel that has a ligand binding spot for IP3. And as a result, calcium, which is built up in the lumen of the endoplasmic reticulum, will diffuse down its concentration gradient into the cytosol. And as a reminder, there are calcium ATPases that help to ensure that calcium ion concentrations are very low within the cytosol. In this way, they are more sensitive to changes in calcium ion concentrations. Now, the presence of the calcium ions within the cytosol results in the activation of an enzyme, and that enzyme is a nitric oxide synthase. And that nitric oxide synthase is going to lead to the production of nitric oxide gas. And it's utilizing arginine that's available within the cytosol in order to facilitate that process. So through this transduction pathway, we're able to generate nitric oxide. Now,